Hello, I'm Jamie Oliver, the Chief Medical Officer for Starton. I want to thank each of you for attending this presentation of the Starton story for lenalidomide. Starton transforms highly successful drugs. We do this to improve their efficacy and tolerability. Required to do this, we modify the pharmacokinetics, which I'll talk about in just a second. The strategy for this is to find new indications or expand the utilization in current indications. Our intellectual property for percutaneous delivery is broad and covers all forms of patch and infusion. We develop continuous delivery systems that are percutaneous of several different types. Patches are derived either by adhesive matrix formulations or polymer patch formulations. For the subcutaneous delivery, we provide wearable pumps or ambulatory pumps to deliver drug over extended periods of time. As I had mentioned before, our plan is to flatten the area under the curve and Cmax and Cmin. We lower exposure up to 90% and can reduce the Cmax, eliminating toxicity, and raise the Cmin, improving therapeutic blood levels. Our lead oncology compound, STAR-LLD, is a form of lenalidomide. We're currently entering phase 1 studies for the compound. Our continuous delivery technologies include polymer delivery as well as adhesive matrix patches. This is a cartoon of a continuous subcutaneous infusion that we're developing. It's wearable and disposable. We also have an ambulatory pump that can be used for longer periods of time and have strong intellectual property, as I mentioned before. I want to take a few minutes to discuss our non-clinical program in rodents. We evaluated PK, efficacy, and tolerability of continuous lenalidomide compared to once-a-day intraperitoneal delivery of lenalidomide. The first study we did as part of our dose finding was in CV17 mice. 3 animals per group were treated. To provide the continuous infusion of lenalidomide, we used a Iprecio pump. This pump is on the left. We treated with a dose of 500 micrograms per day by pulsatile delivery. That's the blue line. In the 4 groups that were treated otherwise, we provided about a similar 500 micrograms per day with the black line. The purple line is about half of that, 288 micrograms per day. The green line is 144 micrograms per day, and the red line is 48 micrograms per day. What we saw in results was doses of 600 micrograms per day caused lethality in about 3 days. We saw some drug-related toxicities at 288 micrograms per day. There were no toxicities at 144 micrograms per day or at 48 micrograms per day. The next study performed in the non-clinical research was a xenograph study in SCID mice where H929 myeloma cells were implanted into the mice. There were 10 animals per group for all of the groups. What we see was the intraperitoneal dose, the blue line, at 500 micrograms per day, which is the standard of care in this model, occurred for 14 days with a day off in the continuous infusion animals to implant a new pump, then another 14 days for a total of 29 days. When we look at the comparability of half-life to expected human doses, we expect the average human dose, or the dose where we saw the most reduction in tumor size, to occur at 9 hours, and the 2 microgram dose would occur in humans around 12 hours. When we look at the efficacy in this study, we found it was quite unexpected, and I'll discuss that right now so that you can understand just how unexpected it was. The endpoint for this study is a tumor volume of 2,000 millimeters, and in the group 1 vehicle animals, or sham, we saw about a 25-fold increase in tumor volume by day 29. In the continuous lenalidomide animals, we saw a 81% decrease in tumor volume, and in the continuous, or in the intraperitoneal dose, group 2 of animals, we saw about a 5-fold increase over the 29-day period. It's important to note that this is a tumor delay model, not expected to produce efficacy, but rather shows delay in tumor growth. When we look at the overall response, we see the intraperitoneal animals that got the 500 microgram once a day intraperitoneal dose had a median time to endpoint of around 47 days, while the total time to failure of all animals was 53 days. There were no responses, either partial or complete, in this group, and no tumor-free animals in this group either, in terms of survival. At 144 micrograms per day, or 6 micrograms per hour, we saw a median time to treatment endpoint of about 65 days, with a time to total treatment failure, which was not reached by 100 days. Surprisingly to us, we saw that there were 6 partial responses and 4 complete responses in these animals at best response, and 2 of the animals survived up to 100 days and were euthanized at 100 days. In animals treated with 48 micrograms per day, we saw a very similar time to endpoint as we saw with intraperitoneal at 41 days. We saw a much better time to total failure of 71 days. We did see a single partial response, but no tumor-free animals or complete response in this group. This is the Kaplan-Meier plot for time to treatment failure in this model. The dashed black line is the duration of treatment, which was 29 days. Then we see that the dose of 48 micrograms per day produced a better time to treatment failure and was very, very similar to the intraperitoneal dose in blue. And the animals that received the 6 microgram per hour infusion had fantastic time to treatment failure. And again, as I mentioned in the last slide, we had 2 animals that were tumor-free at 100 days. The third study we performed was a GLP toxicology and safety study in CD20 mice, which were healthy. There were 3 treatment groups: vehicle control, or sham, a 144 microgram per day continuous infusion, which was the most effective treatment in the efficacy studies, and a 48 microgram per day infusion, which also had activity. The endpoints were at terminal sacrifice at day 8 and day 28. It's important to note that in humans, we would be changing the infusion site every 3 days for infection control. We wanted to identify both an 8-day, which would be a weekly injection, and 28 days, which would be a chronic infusion to assess the key endpoints of local tolerability, hematology through a CBC, and histopathology. As you can see on the right, the vehicle control had 10 males, 10 females, which were split 5 and 5 over 8-day or 28 days, which was similar to both the subcutaneous infusion at 144 micrograms a day and at 48 micrograms per day. The histopathology demonstrated that there was no drug-related toxicity, organ toxicity in any animals, and that the local tolerability in terms of survival, body weight, and infusion site were well tolerated. As I mentioned in the last slide, we wanted to see the 28-day hematology, which is, again, the chronic infusion in animals. You can see from the left side of the figure, we saw, again, another unexpected finding for both male and female animals, where there was no hematologic toxicity associated with either dose of continuous subcutaneous infusion, and similar to sham-treated animals. In addition, all of the hematologic parameters were unaffected, including platelets. There was no neutropenia, and again, no local infusion site toxicity. The conclusion in our preclinical program across all three studies demonstrated some truly unexpected results. When we look at pharmacokinetic feasibility, we saw a decrease in drug utilization of 70% from the daily pulsatile dosing, and an overall exposure or area under the curve decrease of 75% from daily dosing. The Cmax is about 4% of the daily dosing observed with lenalidomide. The outcomes were superior and the depth of response was better. As I had mentioned before, the overall response rate in the animals treated at 144 micrograms a day was 100%, compared to no animals in the active once-a-day pulsatile dose of lenalidomide. 20% of the animals saw complete tumor elimination and required sacrifice at 100 days with the 144 microgram continuous lenalidomide dose, and we had no animals that were tumor-free in the pulsatile daily dosing of lenalidomide. Our safety and tolerability were better than lenalidomide by intraperitoneal injection. We saw the administration of 28 days of continuous infusion of lenalidomide produced no hematologic toxicity in white cells, neutrophils, lymphocytes, or platelets. There was no local toxicity observed at the injection site, no drug-related macro or micro histopathology findings, and that weight changes were similar in all the animals. So the unexpected results that we found were a continuous delivery of low-dose lenalidomide produces better safety and efficacy than once-a-day high-dose lenalidomide. Armed with the animal data, we needed to determine the threshold for both the immunologic effect of lenalidomide, which occurs at lower doses, and the ubiquitination of lenalidomide on Icarus proteins with a direct cell kill effect. Our first goal was to evaluate the immune activation threshold in human multiple myeloma cells for lenalidomide. This study used a cell culture model and was published by Gandhi and colleagues to evaluate interleukin-2 from T cells and interferon gamma from NK cells. What they observed was at a concentration of 0.04 micromoles to 10 micromoles, they saw immune activation by IL-2 release from T cells, and in a similar situation at 0.04 micromoles, they saw active interferon gamma release from NK cells to a maximum at 1 micromole per liter. What we determined from this is that the minimum effective concentration of lenalidomide is 0.04 micromoles per liter or 10 nanograms per milliliter. It's important to note that in cell culture models, they're very similar to continuous infusion in that the concentration of lenalidomide is maintained over the culture period. In this slide, we wanted to determine the published data on the ubiquitination of the Icarus finger proteins with lenalidomide in cell culture. These studies were performed by Cronky at the Brigham. If you look at the Western blot on the left, we can see that at 0.1 micromole per liter, we see some initial degradation of Icarus. And at 1 micromole per liter, we see complete degradation of Icarus. To translate that to blood levels, We modeled a 25 milligram per day lenalidomide dose and the blue line, and see that for some 20 hours a day, the blood levels are below the complete effect on degradation of 1 micromole per liter. And at 0.1 micromole per day, we see that they are subtherapeutic for 9 hours. These data would suggest that the minimum effective dose of lenalidomide is about 0.1 micromoles per liter, which equates to a blood level of 25 nanograms per milliliter, which would be the target blood level for a therapeutic effect of continuous infusion of lenalidomide. It is our intention to exploit the immunomodulation effect of low-dose lenalidomide using a low-dose continuous infusion in maintenance disease. The figure below represents a 7-day therapy of a 5 milligram dose of lenalidomide in blue. In green, it represents a 0.04 micromole per liter concentration of drug, which occurs around 12 hours after dosing. If we look at the effect on Icarus at 0.1 micromole per liter, it requires a blood level of around 25 micrograms per liter. These doses are 60% lower than a 5 milligram oral dose, which should affect both tolerability and survival of treatment. About 35% of patients require either dose reductions and or must discontinue due to toxicity, and the expectation is they will have a better duration of treatment. Based on these non-clinical data, we believe the company is well positioned to move forward into the clinic. I want to introduce Dr. Mohammed Hussein on the board of Starton, who will cover the use of continuous lenalidomide in the clinic.
Thanks so much, Jamie, for the kind introduction, and I would like to thank the organizers for giving Starton the opportunity to present the program on the continuous lenalidomide dosing. One of the things that I thought is critical and to remind everyone is how complex the pathophysiology for hematologic malignancies is, and the different components of it that are influenced in a positive fashion by drugs such as lenalidomide. And I thought myeloma would be a good example to highlight the different aspects of the complexity and the different aspects of the actions of lenalidomide. One of the key areas is the macrophage osteoblast osteoclastic dysregulation, and this area basically is critical in multiple myeloma, and lenalidomide has a positive impact on this particular cellular component. Aberrant stromal structure, and that is critical not just as a support system for the multiple myeloma, but what happens is when the myeloma connects with the stroma, it does become resistant to different chemotherapeutic agents. It does have increased secretion of IL-6 and then eventually self-maintains itself without the need for anything around it to maintain the cellular component of multiple myeloma. That results in a cytokine disruption as well as immune dysregulation in the different components of the cellular system, as you see here, and then eventually result in plasma cell transformation and affects the clonal evolution of the cell, the interclonal relationship, the genomic stability and instability that does occur, and thus the disease progression, which eventually ends in the demise of the patient. Lenalidomide does affect each of these components in one manner or another, and is key in maintaining the disease response And actually, since we do not cure multiple myeloma or cure CLL, it can be a critical component in the maintenance therapy of these diseases. After I showed you the pathophysiology of hematologic malignancies represented by multiple myeloma and the role of lenalidomide in the different processes of the pathophysiology, it would be pretty reasonable to look at multiple myeloma and chronic lymphocytic leukemia for the transdermal approach. In myeloma, the potential for improved efficacy and enhanced tolerability, especially that in multiple myeloma, lenalidomide prolongs progression-free survival and overall survival. In CLL, the immune therapy is lacking, and we have studies that have shown that lenalidomide does prolong disease-free survival. However, though that was compromised in the overall survival because of the myelosuppression and the high infection rate that resulted in death and decrease in overall survival. Thus, lenalidomide continues to not be labeled for chronic lymphocytic leukemia. Here is an overview of the multiple myeloma development program. The first group of patients we are targeting is those patients who are intolerant to lenalidomide because of different side effects. In those patients, we're going to switch them from the oral to the transdermal administration, and we're going to evaluate their ability to continue lenalidomide therapy. We're also going to look at the impact on the depth of response as well as time to relapse. The second group is the maintenance group following cellular therapy. And in this particular group, what we're going to do is those patients who did not achieve deep responses, we're going to switch them to the lenalidomide transdermal, look at enhanced immune response, and look at the depth of response as well as time to progression and or PFS. The third group is the most interesting group, and that's the group of the high-risk smoldering. You're familiar with the ECOG study that randomized patients to observation versus lenalidomide, and the group that got in the lenalidomide has done overall better from a progression-free survival than the group that was observed. That what is important and interesting about this study is half of the patients that received the lenalidomide had to discontinue therapy because of the toxicity that occurred during therapy. And despite their discontinuation, even with a brief exposure, those patients have shown advantage over observation, but were not as well doing as the group that continued therapy to the disease progression. So here, the transdermal should theoretically have a benefit in that 50% that could not continue therapy with the oral lenalidomide. The CLL program is also would be a program that would run after proof of concept in multiple myeloma, and almost to some degree really simultaneously. As soon as we'll show safety, PK, PD data that are mimicking what we're seeing in the encouraging results that Jamie have shown to us, we're going to use the combination of venetoclax in CLL following BTK inhibitors. So basically BTK inhibitor failures with venetoclax plus lenalidomide versus venetoclax alone. We're going to look at the quality of response, and we're going to look at the progression-free survival. That would be a new indication, as Revlimid is not labeled for CLL. We're going to look at combination with BTK inhibitors in high-risk CLL. This is a earlier kind of patient population. In this one, basically, we're going to try to look for a biologically active dose rather than a maximal tolerated dose, and then we're going to assess PFS, MRD status, and then assess control versus BTK alone. So we're looking the same quality of response in PFS. Now that I've shown you and discussed the clinical program, here is a timeline where towards the end of the quarter of 21 would start a phase 1A in Europe. First, second quarter of 22 start phase 1B in the United States to expand indications in myeloma and initiate CLL indications. We're looking at orphan designation in CLL, potential breakthrough in CLL, and actually in multiple myeloma. In conclusion, lenalidomide in the management of multiple myeloma is a critical drug. However, though, there is a group of patients that its use is not optimized and the patients do not receive the full benefit because of its side effect profile. Also, immune modulation is a cornerstone therapeutic approach in the management of CLL, and further development of lenalidomide with reduced toxicity and improved activity is warranted in this disease area. Again, I would like to thank the organizers for giving us the opportunity to present to you the preclinical and clinical programs with the timelines and the rationale that's involved in that, and we're more than happy to take any questions. Thank you.